Okay, so good morning everybody, and thanks a lot for the kind introduction. It's a pleasure for me to talk to you today about our developments we are doing in the arena of cell culture media. Maybe not everybody is aware that we have digged into that already. We have developed recently some proprietary CHO formulations, so I will show you some data on that. But what, from my perspective, responsible in Darmstadt for the transfer into large-scale operations, into production, the dry powder, I think it's really important to make sure once you have developed what kind of platform we have talked about in the last couple of days and maybe also today. I think it's important really, if it comes to the point of large-scale supply, to make sure that you have really the consistent quality, and I will show you what we are doing in Darmstadt and how we address that to make sure that besides the formulation, which is only one part, we can deliver the right quality. So therefore, I would like to dig in first, let's say, into the recent development ending up in the own formulation. Then I will show you some lot-to-lot consistency data, platform consistency from a powder perspective. Then I think it's very important that you try to figure out as early as possible if there are some trends in quality shift on your end, and you're the owner of the process and can probably much better judge about all your critical quality attributes of your molecule. But we will address from the very beginning on, let's say by doing media prediction, if we could support you in identifying critical things going on maybe in the dry powder supply, and I will show you some data how we address that. Powder handling improvements. We see, let's say, in the industry coming up with some fancy recipes with increasing concentrations maybe of hygroscopic molecules, and how do we, let's say, treat them? How can we handle that problem if it comes to powder transportation in large-scale operations? You need to make sure that you have on that end also really good, let's say, powder properties to make sure that you can achieve, let's say, the desired endpoints. Also, let's say we have talked a lot on the media platform, and a part of the media platform is a feed strategy. I will show you some developments, what we are doing to make sure that you can simplify your processes by, let's say, allowing to transfer probably the most critical components, the tyrosine and cysteine, into the main feed. And therefore we have done some developments, and I will show you in a second more details behind this, and I will then summarize with some of the perspectives. Yeah, coming to the own development, and as I mentioned before, so this will be commercially available by early April, and if you're more interested in, so please join our colleagues at the booth, Merck Millipore booth in the exhibition area, and they can discuss this more in detail with you. But what you see here, let's say, if we are doing, let's say, with regard to the competitor platforms, let's say the check on viable cell density and also on titer, we really can come up here with a very nice product for both ends. Of course, this is clone dependent, and as we have heard over the last couple of talks, cell line is not cell line, and this needs to be tested in your hands. But we are really, let's say, satisfied with the development so far, and I think this could be a good starting point. And then from that end, we can support you in, let's say, also try to adapt this to your needs if you're interested in moving forward. So this is not the end point that we only hand over a product to you and say, oh, come on, it's up on you to develop, let's say, the process with a given formulation. We also can support you in doing fine-tuning according to your needs. So if this is of interest, so let's say, let's discuss this in more detail later on. Coming then, let's say, to the competitor, so the abbreviation is called Solvento CHO 200. So this is our platform. There is a media and feed, and then we have compared this, let's say, to competitor media and feed supplements. And here you see the VCD. It's quite nice for that type of, let's say, cell line. We are ending up somewhere at 1.2 10 to the 7 cells per mil. And also you see here, let's say, that after day 7, you have somehow not really the full immediate decrease in viability, so it's a decent formulation. Lot-to-lot consistency, and that's more, let's say, something you should also on your end, and I am sure you do so, have a closer look on the powders as such. Let's say what we are doing here is, let's say, of course, see once we have produced 3 different lots of powders, that we have the consistent quality. Therefore, you need, of course, to set up the right experimental conditions. But what you see here, let's say, only, let's say, for the cell growth in batch culture, you can see immediately. So there is a pretty nice, let's say, correlation and a pretty nice consistency over the different media powder lots produced so far. And if we then digging into the comparison of the feeds as well, so also you see here, let's say, that for the fat batch process, you have very similar conditions, at least up to day 11. So then you see some deviations over here, but let's say, so the overall consistency looks nice from the cell biology perspective so far. Then if we compare, let's say, the whole platform batches with regard to titer, you also see here that based on day 7 and day 10, you have really comparable and lot-to-lot consistent quality over the 3 batches. And why I'm digging into that is if you have, let's say, some of the formulations, and we are talking about trace elements, and maybe trace elements are driving the performance of your processes, and I'm sure some of the people are really knowing what I'm talking about, is that, let's say, it's really important to make sure that you have the precise amount of trace elements in your formulation, and therefore it's mandatory also to understand the overall impurities coming along with the other ingredients. And let's say if we're talking about impurity profiles, we can proudly say that we have really the grip on all the molecules with regard to the contribution of up to 60 different impurities which can come along in the media formulations. And to give you just a number, so in a kilogram powder, does anybody have an idea what we are talking about with regard to impurity? Is anybody here having already digged into that? Are we talking about, let's say, milligrams, grams, or what is a let's say the contribution of impurities in a media formulation coming along with amino acids, sugar, or sodium chloride? Normally, everybody should know this because I think this is a real problem we are facing, let's say, in the media industry, and we are digging into that. And just to give you a number, so we are somewhere around, let's say, the 15 microgram per kg, and we know what kind of molecules are contributing to that. So this is really important understanding. And if we now go back to the, let's say, powder production, you have really to make sure that you, if you want to, let's say, adjust exactly selenium maybe as one of the components or some of the iron salts, that you have not really, let's say, larger amounts due to the fact that you have some impurities coming along with the glucose. So that's a point I want to make, and there we are really strong with our 300 colleagues in Darmstadt supporting us in all kind of analytics and in the release of the individual components. And that makes us confident to show you here the data that we have really the batch-to-batch consistency in the dry powder lots. Then we can also, let's say, correlate this with regard to physical-chemical properties. So it's important, depending on, let's say, what is your exact need, and of course, with regard to your formulation, to come up with the right powder. Not always downmilling to the lowest particle size is really the desirable approach, because depends on the amounts of hygroscopic molecules. You have, let's say, lower powder flowability, the more you mill down the overall formulation. You need to understand that within the Nernst equation for dissolution rate on one hand and the physical-chemical properties on the other hand, you can play the card of adjusting the particle sizes, so that you can come up really with high concentration in solutions. And that's something where we work heavily on in Darmstadt to really make sure that we can come up 140, 150 and even higher, let's say, gram per liter of media and concentrated media and feed formulations, and to make sure that we can bring them in solution at close to neutral pHs. And here you see an example of the particle size distribution curve of 3 different batches which we have seen so far. Also here you see it's really a nice equal distribution, and it's a nice overlay of all the 3 different batches independently produced. Here you see the same story for feed, and there are 3 lines lying above each other, so it's really also here saying we have, let's say, full control over the particle size during our milling and blending process. If it then comes to amino acid analysis, reproducibility over the batches, also you can see here we have this adjusted, let's say, to the 100% as is in the concentration of the media formulation. And you see for the different amino acids, let's say, also here you see over the 3 batches produced really nice confident data showing that. Now, platform consistency. So you would also, let's say, then ask, okay, how can we transfer this from the, let's say, TPP approach, the 30 milliliter mini bioreactors, to larger benchtop bioreactors? And also here we have the 3 liter cell ready, and you see also here, so it's very comparable data with regard, let's say, to cell growth in spin tubes versus the cell ready 3 liter bioreactor. And if it comes, let's say, then to titer, also here you can see, let's say, very comparable results on day 7 and day 10, let's say, in these 2 setups in here. And now I'm coming to what I have mentioned already in the very beginning, so the media performance prediction. I know that not everybody is willing or able, let's say, to share the clones and the processes, and you own the processes anyway, and I think you drive them much better than we can. But what can we do, let's say? If, let's say, we would provide you with some kind of media samples, which we have produced in different ways, you just go back, let's say, into your laboratories and do the testing. And the only information we need, let's say, is if it's a good, medium, or low performer. And then we will do the principal component analysis using different type of analytics. So we can do NMR, NIR, MIR, Raman, what else? And then we will find out whether there is a trend to cluster these, let's say, type of performance indications given by you. And this is just an example, a simple thing. And not one technology works for all, but we can find out together what is the most appropriate, let's say, technology for you. And therefore what you see here is, let's say, we have here produced different type of media batches and then run through the whole process. And then you see, of course, is a correlation between a simple NIR technology, a simple part of the spectra, which can be principally component analyzed, let's say, with regard only to the 3 endpoint media, high and low performance. And you see there is a pretty nice, let's say, a ballpark. And what is the rationale behind it? So if there is now in the future a shift, let's say, if we have adjusted our processes in here, and we see a shift to low or medium performer, we can immediately, let's say, react and say, come on, something is going wrong in any kind of direction before you run through the full process in your laboratory with all the labor-consuming, let's say, bioapplications. So this is the intention, really, and therefore we don't need to own your processes, but we can offer you to work close together in here. And this is just an example, let's say, let's say, more spread out. So if you then cluster, let's say, different type of DMMF12 media, which have produced by various suppliers, so that we really see, let's say, there is a trend, let's say, in increased performance coming from the left upper hand side to the right hand side down here, so that we really can see, okay, we can produce different batches and always ending up here and showing that, let's say, even this is not a high-performing media for sure. But just to give an example and to talk about a recipe which is known to everybody and where you can get immediately dry powders in hand to compare this, so that there is really something behind this that you can use this in performance prediction. Now I want to dig into another point. We have heard yesterday a lot about the glycan patterns. So seems to be very important that you have a look on the critical quality attributes as such. So, but therefore it's mandatory to have also the right analytical setup in place before you even start thinking about downstream, let's say, purifying, analyzing, and what else. So what we have done here, and this shows you, let's say, how important, of course, all the formulation work is you're doing, but besides that, you can kill even all performance if you don't produce the powders right. And I show you why. So here we have 2 types of mimic technologies, called 1 and 2. If you run, let's say, a simple test, and let's say only look on the population doubling levels for CHO cells here, for a certain type of media, so what you see basically is there is no big difference between the 2 types of produced powders. And so far, if you dig not into that more in detail, you say, come on, it's fine and I'm done and all things looks the same. But if you have a little bit more a closer look with a more sensitive assay, so the cloning assay, limited dilution assay, you see that there is, let's say with the milling technology 1 compared to the milling technology 2, a tremendous difference. And we are talking about the same subset of raw materials. So we have used the same lots of the raw materials, and then we have produced either this way or that way, the same formulation, and then go through the same biological assays. And there you see there is a tremendous difference. So that's what I meant with the fact that, let's say, it's not only important to do the formulation work, but also to do the proper preparation of the dry powders. This has also an impact on titer, but this is not the point I want to make, because you see here a slight increase in titer, maybe 10 to 15%. But I think with regard to the discussions we had yesterday, it's more important to show the, let's say, relevance to bridge from the raw material part, producing the powder, and what is the impact of critical quality attributes of a molecule. So this is shown in here. Here, let's say you see the acidic peptide group. So it means you trypsinize an antibody, you cleave it, put it on a column, and elute, let's say, the neutral, basic, and acidic fraction, calculate area under the curve. And this is part of the filing for, let's say, for the authorities, so that you have, let's say, data on hand showing, okay, for this specific antibody, the acidic peptide group concentration is, in this case, for some of the molecules, let's say 34%, and then let's say you have an internal specification saying, okay, 30% to make sure that we have enough space, security space, but you see that you come close to that limitation, and then you have to reject the batches. And nobody in the industry is really talking about how many batches they have to reject in the industry, but the given is a fact that there is a certain number of batches, and I think all the large-scale producers are interested in reducing that and getting a grip, especially for the monoclonals, on such kind of, let's say, analytics. And the idea is, if you could already, by dry powder formulation and milling technologies, lower the level, because this has also contribution of the molecules, stability in patient samples. So there is really a medical need behind that, not only the economical, so that you can come up to a target space, let's say below 25% roughly. And we could show that we can produce the powders, and we have a direct impact on the critical quality attributes of the final molecule. Okay, so powder handling improvements. I mentioned before, so we see a little bit the trend that the concentration of hygroscopic molecules increases with the formulations people are developing in their labs. And what you see in here is a simple powder, DMMF12. So the overall concentration is 1.98% of hygroscopic molecules in DMMF12. And here you have another chemically defined media. Looks nice, so the concentration here is similar range to the DMMF12. But here we have another chemically defined media, and you see immediately, if you do not take care of producing this properly, you end up with a clumpy powder. And if you do a simple analytics like particle size distribution analysis, you can immediately see that after a couple of minutes, 3 minutes, let's say, in an airjet sieving, you have something like popcorn, like chewing gum. So just with this kind of formulation. This shows you that the control of humidity and, let's say, handling of powder is really important to make sure, let's say, that later in large-scale operations you don't run into any kind of problems. So by doing the root cause analysis, what was the reason that one or the other is, let's say, more hygroscopic? If you just add, let's say, these numbers together percentage-wise, you see, let's say, that for the chemically defined media, one, which we have seen, which looks nice, comparable to the DMMF12, we end up only in a concentration of 1.5%. Whereas in the other combination, let's say you see 9.7. So this is a 6.3-fold enrichment of hygroscopic molecules. And this you can already measure by Carl Fischer titration. You see the increase immediately if you do not protect, let's say, the powder during production, that you can easily end up at roughly 4% Carl Fischer. And this is an indicator, of course, that you would expect a clumpy powder, not a free-flowing powder anymore. And I will show you why is humidity somehow, let's say, important. Normally now these small movies should show up. Can we fix that, or? This morning we have done the dry run, it works, but for any reason?
It's got humidified.
Sorry?
It's got humidified.
Yeah. Okay, so but if we can't fix that, sorry for that, but what I want to show you is humidity is, let's say, in so far interesting because this has a direct impact on solubility. So because the solubility, let's say, you would expect, okay, if I would extract some water, would be nice, let's say, to support, oh, now you can see it. You see some granules in here. This is DMMF12, let's say, just treated with certain humidity over a certain time period, and you see immediately this is already in solution. This is an accelerated movie over 5 minutes, down scale to 1 minute, and you see immediately here is things are going on. Why do I tell this? Because let's say with the hygroscopic increase of molecules in the formulation and not taking care on that, you will run into solubility problems. And the more you're interested in, let's say, high concentrate media formulations, this is something you need fully control over, otherwise really you will end up, let's say, immediately with some precipitates. Because if you start, let's say, with this type of media formulations, and this is of course an extreme example, but what I want to show you, if you now increase the concentration of all the raw materials in some, then you have, let's say, the environment allowing, let's say, hard to dissolve components immediately to precipitate. And this is something you need to understand. And that's, let's say, is a part of research we are doing in Darmstadt on powder properties. Not on the formulation work, but really trying to understand what is behind the powder. Okay. Good. So thanks that you have fixed that. But now we need to go back to the presentation mode, please. Excellent. Thanks. Okay. Good. Yeah, so there's also some improvement from my end to make sure that we can plug in the movies much more smoothly. But now what we are doing is also, let's say, to understand and measure, let's say, the powder flowability, which is coming along with the hygroscopic increase. So we are doing, therefore, let's say, the powder analytics by calculating the break energy and the avalanche angle, and this is something you can immediately see. So if you have a clumpy powder, this is lifted up in a rotating wheel, and then depending on how clumpy this is, it drops down at a higher point, and this, let's say, line is in, let's say, gives you the avalanche angle, at which angle, let's say, the powder tends to flow back, let's say, to the bottom of the rotating wheel. So that's basically something you can see beside the immediate differences, let's say, in changing color. And in so far also here we are strong in, let's say, finding solutions to make, let's say, powder analytics. And what could be then some option for you to improve the powder flowability? This is roller compaction. So if you now have a formulation worked out over months and months and months in your laboratories, you want to make sure, of course, that we can produce them in the right way. And therefore we have, let's say, also then the possibilities to do the roller compaction. And here you see some nice fancy granules where you can adjust the particle sizes also of the granules to your needs, depending on, let's say, your powder transportation systems. And what we have is, let's say, depending on the different pressures, we have applied different, let's say, molecules, and you could see them here under the microscope. So here has the uncompressed powder, and then the increasing concentration of pressure with particle sizes ending somewhere up between 1 and 3 millimeters. So this is something which then has a direct impact on the powder flowability, because then you reduce, of course, the Carr indice and the Hausner ratio, which are indicators for powder flowability. And then you would expect to ask me first question, but what about solubilization of this type of molecule? So is it something which needs, let's say, longer time? No, because normally you see always the same problems, that some of the amino acids are floating on top, and before you bring them in solution, it takes quite a while, up to 10 minutes, whereas, let's say, the granules immediately sink down and can be then, let's say, steered from the very beginning more efficiently, and in so far you end up at a very similar dissolution time. And we have also done some analytic on the effect of, let's say, the performance of the media, and also here we can say that there is only a little or no, depending on what kind of endpoint measurement you are doing with regard to performance, but this can give you the opportunity to have have dust reduction in GMP environment, have better powder flowability. So this is something we need to discuss case to case depending on the formulation. Also here, let's say media supply is not throwing a recipe over the fence and say, come on, producer produces. This is an interactive approach. You need to talk and to understand really what is your need, and then we can adjust to that. Part of, let's say, filtration, we're coming closer. Okay. Filtration, we are also investigating and give you some guidance on possibilities how to filter media. Let's say also here we can discuss more in detail. But what I want to show you as the last, let's say, innovation we made in Darmstadt is increasing solubility. So here we have already a tyrosine molecule in our hands, allowing us, let's say, in a feed already having over 110 gram per liter of other molecules, let's say, solubilized, to start here with the L-tyrosine. You see with 1 gram per liter already, let's say, precipitated. Even the disodium salt of the L-tyrosine is precipitated, and then we have modified tyrosines, which you can use up to 70 gram per liter in your main feed at neutral pHs. And we are having, let's say, in the meanwhile, worked out a scenario where you can have a full media and feed platform, let's say, making the L-tyrosine and cysteine in certain concentration redundant. So this is also something we can offer to you. Good, and here you see, let's say, just one example of comparison of tyrosine disodium salt, which is a current gold standard in comparison to our molecules in different, let's say, concentrations. And also here seems, let's say, that, let's say, from the growth perspective and also from the titer perspective, and it looks nice and has no impact on the final molecule. Also here we can share data a little bit more in detail if this is of interest. Okay, so by having said that, I think I summarize, and I think hopefully I could make the point, let's say, that it's really important to have the grip on the raw material quality on one hand, and that you need to understand the production process of the dry powders, not only the formulation, which is a very important part, of course, but keep in mind, let's say, if it comes to constant supply and to batch-to-batch consistency and performance prediction, also something we can discuss more in detail, and therefore the milling, mixing, and the packaging concept is important, and the combination of what I've mentioned before is really key to support you with the maximal, let's say, product quality. And by having said that, thanks a lot for your attention. I'm happily looking forward to take some questions if you have.